how to. I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas at San Antonio's Neuroscience Research Podcast. It's October 8th, 2020, and today we welcome Michael Hasselmo, who is William Fairfield Warren Professor of Psychological and Brain Sciences and the Director of the Center for Systems Neuroscience at Boston University. Hi, Mike.
Hello.
Mike's research takes an interdisciplinary and multi-scale approach to memory, from neuromodulation and firing patterns of single neurons and networks to computational systems neuroscience and behavior. His current research focuses on the neural mechanisms of spatial navigation and episodic memory, specifically how space and time are represented in the hippocampus and related cortical structures. So around the table today, or well, in the Zoom today, as you can see, we've got a group of physiologist modelers, fellow navigation specialists, Isabel Muzzio. Hi, Isabel.
Hello.
And Francesco Savelli. Hi, Francesco.
Hi.
And of course, we've got good old Charlie Wilson. Hi, Charlie.
Hello.
So Mike's work in the memory field is so fundamental and pervasive. I think it's most appropriate to point our listeners to his 2011 book, How We Remember: Brain Mechanisms of Episodic Memory, for a rich account of how the brain records and integrates the spatial and temporal features and experience as trajectories on which to connect meaningful objects and events for later retrieval. We also, I also want to point you guys to the fact that we have in our archive a 2016 conversation with the late great Howard Eichenbaum, who first introduced us to multifunctional space and time representations in place and grid cells. And that work that he discussed in that podcast, which you can find on our archive, was a longstanding collaboration with the Hasselmo group. So for today's discussion, I think a cool starting point is how biophysical and network models have supplied the parts list for understanding how the brain codes space and solves navigation. So Mike, it seems like your models have been particularly prescient in driving discovery. So before we knew that there were cells in the entorhinal cortex that represented a time dimension, the models, I guess, told us that they would be there. Another example is your model for how time might be represented in a network predicted exponential time decay cells. So also now more recently, I guess models have predicted that boundary sensitive representations are necessary for coordinate transformations between egocentric and allocentric space. And you found those cells in the retrosplenial cortex. So that's a lot of data that I hope we get to bearing out a bit of, but just in a very general way, taking a step back as a biologist modeler, because first and foremost, you are a biologist asking biological questions. These are not toy models. Can you say something about how much the models drive the experiments and how much the experiments, for example, the behavior work that you're doing on context, for example, how much those maybe drive the models and your take on some of that?
Yeah, so it's nice that you're highlighting the models, because I am very interested in the models, and I always kind of am operating with working models in my mind when I'm thinking about new experiments and really trying to understand how the system works. And my overall goal is to have some kind of comprehensive model of the mechanisms of episodic memory, but even of broader cognitive processes that are involved in regulating when to store information and when to use it for planning and so on. So I really am coming from that perspective, and it's very exciting, when models make predictions that are then borne out with experimental data. You mentioned already the one about the transformation from egocentric to allocentric representations. I think Neil Burgess deserves a lot of credit for that very exciting model, because his earlier component predicted the allocentric boundary cells, and then this model then went on to predict the egocentric boundary cells that have come out just in the last couple of years. So that really is, very exciting to see those sorts of predictions bear out in the field. And the time cell, you could even say that Tulving's definition of episodic memory in a way predicted the need for time cells. But I think the grid cell model also kind of generated this possible function of mapping dimensions other than space, namely time, So really there's a lot of potential for the models to guide the experimental work.
You say something about how to make models that make predictions like that, because a lot of people have the notion that nothing can be in your model that you didn't put there. So if you didn't know it existed, it wouldn't be there, but somehow you can put things in your model that you didn't know you needed. Or
yeah, and I think it comes out from the process of completing the model. So a lot of people want to work with sort of verbal hypotheses, and I think a lot of neuroscientists do that. They have an internal hypothesis that's kind of a word-based hypothesis, but the process of actually programming a model and putting it together puts a lot of extra demands on you to make things fit together. And I think that's really where a lot of the new ideas come from. So, for instance, the work by Neil Burgess, he was trying to model how play cells changed when you move the barriers in the environment. And I think it was the process of trying to build that model for that really one experiment that forced him to come up with the idea of these allocentric, boundary vector cells, and then the egocentric boundary vector cells. And I, when I was trying to address the role of the theta rhythm and kind of the potential encoding and retrieval dynamics, I was trying to model the kind of reversal, learning of reversal, and could when addressing what the criteria were for behavior in the context of theta rhythm oscillations, I could come up with what the phase relationships would need to be, and that actually made finer grained predictions that still haven't been fully tested about the potential firing phases and the phase relationships on a finer grained basis.
You mentioned velocity there, and so it's interesting. There's also this idea that velocity is coded in two different ways, I guess. Is it in the retrosplenial? There's a theta rhythmicity, which you found, and then there's
that was in entorhinal cortex.
In entorhinal, sorry, and then there's a traditional linear rate, firing rate, also. So when you find something, and you've got your part list, and you found the velocity input, and then you've got this sort of bifurcation of signals. As a modeler, what happens then? I mean, do you go back to robotics? Do you go back to engineering? Or is that where the model ceases to be, and you sort of keep looking for more connectivity, maybe biological realities? Or...
Yeah, I mean, in this case, this is sort of different ways of approaching the same problem. So, you have the representation, the need for representing velocity, and then you realize that there's different ways of representing it with the firing rate changes or with the theta rhythmicity. And I have to say, I'm on grants with robotics components to them, but roboticists are not making these kind of biological distinctions yet. Maybe somewhere down the line they will, but they just, they'll go for the kind of the quick and easy algorithmic representation. As soon as it works, then they're not worried about implementing it. Maybe if they had to put it into neuromorphic, circuits, then they'd have to start dealing with whether it's represented with oscillations or, some kind of analog signal. But really it comes down to kind of starting to match the data, and, then you can get into specific analyses of the experimental data to see which mechanism works best, and you find yourself often getting even more fine-grained representations. I think I was talking with somebody today about the fact that you really can represent velocity in terms of the influence of optic flow as well, and that could be either firing rate or rhythmicity, but it could also have other dimensions pertaining to the way that the optic flow is represented. So you really start to kind of almost have like a branching of the models into more and more experimental options.
Ask you a question. I think that perhaps in the field of navigation and memory, one of the biggest challenges, and your lab has been an exception because you have been addressing to some extent what I am going to ask, but is that we are trying to interpret how our sensory system is integrated in the phenotypic firing that we see in these different cell types that we observe in the medial temporal lobe, and how they generate the different rhythms that happen in vivo. But at the same time, we need to understand the intrinsic firing properties of the cells that are part of those circuits. And that has to be done in vitro, which presents a challenge. So your lab has been different because you have addressed both questions, but how can you integrate both types of information? Because without that integration, it's going to be almost impossible to understand how you give rise to a grid cell, for example, if you don't understand the intrinsic properties of those cells in terms of ion channels and how they fire. And at the same time, without the in vivo component, is very difficult. So how do you integrate all that in the models?
Yeah, and that's that's a central question for the entire field, not just for me. And I've wrestled with these problems. We've done slice physiology, we've done biophysical modeling, we've done in vivo recording, we've done modeling of the in vivo data. And I have to say, I find it frustrating that there's still different levels of modeling that are quite distinct from each other. So you can, and this is partly in my own work as well as in the field as a whole, there are the biophysically detailed models that can address the spiking dynamics of the memory potential of a single neuron and the currents and conductances that are contributing to that. And then there's these functional level models that address the in vivo data and the behavior, which are to date almost always more abstract. And bridging between those levels is extremely difficult. I did that early on with biophysical models of attractor, associative memory networks, not really attractor networks, but with, biophysically detailed models. And we modeled the phenomena like persistent spiking and match enhancement, things like that in biophysically detailed models. But to take a biophysically detailed model and map it up to the in vivo data is very challenging. And we've come close. Eric Zilli did this with me in a model in 2010. And Dan Bush did a model with Neil Burgess in 2014 that at least had spiking dynamics for a grid cell model. But both of those models were still relatively abstract spiking, rather than full biophysically detailed models. So this is something that it's a tremendously complicated process to get working. And I'd like to see more people doing it, but if anything, I have the feeling there's fewer people trying to make the, the mapping.
I was thinking about exactly that today when you presented the data about the projections from the septum to the entorhinal cortex and the new results in which, you showed that, if you lesion the septum, people have shown that memory is disrupted, and then you also show that eliminating the projections, the grid cells disappear. And then you show that more recently you have been looking at cell specificity, and you, if I recall correctly, you said the GABAergic input appears to be the one that mediates the grid cell characteristics. But I was remembering there is a recent paper by Zomogi that says that it's a particular type of GABAergic cell that projects to it's a type of parvalbumin cell, because apparently there are many parvalbumin cells that project, but these are specific parvalbumin cells that have a particular firing pattern. They fire in bursts and they have these characteristics. So I was thinking whether it's a more general problem in the field that those that specialize in looking at the intrinsic properties of how the cells fire, maybe there is not enough crosstalk with all the people that do the in vivo work to put together the big picture of how, the brain is working in a particular region.
Yeah, I agree. There should be more interaction. And Samoji has been great in the sense that he does intracellular recording in behaving animals now, getting the theta rhythm dynamics, and he can look at the spiking of the neurons relative to the network theta rhythm oscillations, and Klausberger as well is doing that. So that's exciting. And they're starting to make those links. But I have to admit, most of the people doing extracellular unit recording are not thinking in those terms. They're not trying to connect to the Samoji data or the the intrinsic properties, a lot of them aren't thinking in terms of spike frequency accommodation at the single cell level or other intrinsic properties. We've done work on the H current, and we had a whole project about how the H current could be involved in grid cell generation. And that was, exciting. At one point, I thought that would be kind of a simple, clear story, but it ended up, as often happens, being more complex than that. But it is a great opportunity when you can make those connections.
I think one difficulty in bridging the two worlds, especially at the modeling level, is that there is some sort of mismatch between the complexity, or rather the detail of information that you have. So you might make a very biophysically detailed model of the neuron, which is great, with all the dendritic compartments and ionic currents and everything, but then you don't know enough about, how the patterns of inputs, say, if you're modeling, I don't know, grid cells to place cells or whatever, you don't know, okay, how many inputs do you really get? Or, how many cells are really with these characteristics or not? Because, there are estimates in the field, but they're very rough and they're subject to sampling bias. As somebody who's done those kind of experiments, there's always an inevitable degree of sampling bias. So you end up with a model that it's kind of lopsided in a way that is like very detailed in a way, but then that detail doesn't match other details that you need. And so you start questioning, well, maybe I'll make a model that is more abstract and hopefully I can capture some kind of principle, and so on and so forth. So it's very important, but it's also very difficult for that reason, I believe.
Yeah, you laid that out really well. That kind of it describes exactly the evolutionary process that a lot of people go through when they're starting to do modeling, especially if they start at the biophysical model and want to go to the more behavioral and in vivo kind of level, that they'll start with some standard biophysical model, and then they try to put it together into a population and link it up into a behaviorally functioning circuit, and immediately they run into that question: how many of these connections do we have from this region to the other region? And even within a region, what's the percentage connectivity? How is the function associated with those percentages of connectivity? And you end up guessing at so many of the parameters that you feel like it's all hypothetical. But at the same time, the process is useful for people to go through, because they realize it kind of, opens their eyes to how much is not known. It's stuff that people doing just unit recording don't even kind of think about. Oh, well, there's these problems of how the different cell types are connected and what their intrinsic properties are, all the things that you have to guess at when you're putting together the models. So on a certain level, it's useful for making you realize, just the huge number of questions. I actually wrote a paper recently called The Unexplored Space of Neural Models that was trying to address this specifically, because my sense is that people tend to cluster into particular types of models, like the attractor dynamic model, which is very popular, or else the feed forward kind of deep learning trained, gradient descent trained models. And if you try to think about the whole space of possible models, those are only two really restricted areas of this parameter space. And there's just this humongous domain of unexplored types of models that you could create if you actually took into account all the possible intrinsic conductances and all the possible patterns of connectivity. And, I'm thinking that maybe we should try to explain what hasn't been explored. I had a map of, from I think that, right when Magellan was doing his circumnavigation of the world, showing what was known of the world and how the whole western coast of the, Americas was completely unexplored, but at least they knew what was unexplored because they had longitude and latitude. They had some way of quantifying what was explored and what was not explored. And if we could even start by having some, description of the dimensions of neural function and figure out how much of the space is unexplored, and then maybe send people out to explore these different domains of modeling space, instead of just lumping into the attractor dynamic space or the feed forward gradient descent space. I think that could be a way of approaching the issue.
I agree. And there's even more than that. Sometimes even the terminology becomes in a sort of Orwellian way limiting because, for example, one thing I always thought, so there was this dichotomy between, attractor models and oscillatory models, which you know very well because
Yeah.
a lot. But for example, what people intend as attractor model is a very narrow implementation of the concept, the very general concept of attractor in physics, right? So it's like a firing rate idea of this network in which each point has a firing rate. And so you have a vector of firing rates for the population of cells, and that's your state space. And then the attractor is a point in that state space or is a line. But that doesn't have to be like that. I mean, you can have an attractor dynamics in an oscillatory space, for example, and it's called synchrony. You have a network of cells that, converge to synchronization. You can think about that as an attractor dynamics.
Yeah.
So sometimes even just even the terminology itself, I feel like sometimes it's kind of constraining and limiting the way that people are thinking about it. So that goes, to what you're saying.
Yeah, it's true. I mean, I should have said fixed point attractor models because there's all sorts of limit cycle attractor models, and frankly, they're in the unexplored domain.
Yeah. But but yeah, I mean, in general, I agree with what you're saying. I think you put it very well. The model, so coming from computer science, we are saying programming is understanding, and it's so true. That's the idea. It's like we all have verbal models in our mind, otherwise we wouldn't be doing research. The problem is that you have this verbal model works great in your mind, and then you try to implement it, and all sorts of things come out that you weren't thinking about, and it fails in all sorts of ways. But I just feel like, that's when you're really usually learning something, at least as you said, you learn what you don't know. And occasionally the model actually points you to some kind of insight because it behaves in a way that you weren't expecting. It worked, you just had, you thought you had it all figured out in your mind, and then you did, you actually didn't. And that's actually going back to what Charlie was saying. Yeah, a lot of people think, well, you put it in a model. There's this joke saying, a model is doomed to succeed. Right? And it's true in a way, and everybody's done modeling knows that. At the same time, no, that's not true. I mean, oftentimes what you think you're gonna make succeed, when you just implement it, just fails. And you weren't expecting that it failed. And why does it fail? Oh, okay, I figure something new. And so it's actually an extremely useful exercise to do modeling for this reason.
Yeah, and I've encountered that, many times when I think that I have this intuition that something's going to work in a certain way, and then I try to program it and I realize, oh no, yeah, I was forgetting such and such feature that prevents it from working that way. So I have found that useful. And I know, I do know this phenomena that, a lot of times, well, frankly, what Charlie was saying about the modeling addressing the existing data, part of that is that even being able to design a model that addresses existing data is a hard process. And so people often will start with the goal of, making a general theory that's going to make predictions, and then after many years of work, all that they've managed to do is to account for a subset of the data. And so I would be excited even if models could account for all of the existing data. That would be just linking together, for instance, the cellular level intracellular conductances and the network level in vivo data. If we had models that could just address the existing data, that would be a pretty amazing achievement. But it is, the ultimate goal of models is predictions. And I know that, for instance, in physics, everybody's criticizing string theory because it only is addressing existing data and not making new predictions. And that's, the real gold standard of models is when they generate new predictions. But that's out there in neuroscience. The Hebb rule was floating around for many years before, even before Bliss and Lomo, before LTP was found, and certainly before the Wigstrom and Gustafson and McNaughton and so on, showing these Hebbian properties of long-term potentiation. So that was a success of modeling even though it doesn't maybe get emphasized as much as it could be. And there are these other examples like the boundary vector cells. I mean, that's sort of an unsung achievement, because that's quite an amazing prediction that turned out to be supported much, much later by experimental data.
Yeah.
So I know this is a little off topic, but one of the things that strikes me about your field is what's happening with basically with place cells and grid cells literature. So it started out, as I remember it, there were place cells and they were in the hippocampus. And then, and they have all these interesting properties. Maybe they tell us where we are. And then the question was, where do they get that information? And it kind of grew out to entorhinal cortex, which was a natural place, being kind of the immediate upstream place for the hippocampus. And now listening to you, I'm getting the impression that this system is growing. It's going to soon have taken over the brain. So it's not just the entorhinal cortex, but there's the septal nuclei, and there's now cortical regions and basal ganglia regions. They're all part of this system. So is this a sort of success story of like chasing the place cell and gradually constructing the entire system of the forebrain? And
Yeah.
Is this going to work?
Well, I mean, it seems to be progressing at least, so it seems like science needs these little seeds of really hard and fast facts that are highly reproducible, so that was kind of the appeal of it, was that the place cell was such a robust phenomena that other people could go in and really robustly reproduce that phenomena and build from it. So even though people would often criticize place cells, they'll say, well, why study them? They're so far from the sensory periphery, so you don't know what's causing them. So that's a kind of a standard, argument for people working in primary cortical areas, that you shouldn't work on place cells. But it's exciting, and it really is, just because within the relatively constrained foraging task that it started with, at least it was highly reproducible, and therefore lots of groups could go in and see the phenomena and then link it to other things. And it's kind of, it's actually kind of amazing how long it took, the interval between the 1971, the first place cell publication, and then the 2005 grid cell publication. That's a pretty amazingly long interval, especially considering that people knew that entorhinal cortex was providing the input and they looked there, so Quirk was looking in entorhinal cortex specifically for inputs to grid cells, and other people were looking many years ago, and it really wasn't. It's interesting because it's kind of an interesting story, because the problem was that they were all recording in ventral areas of entorhinal cortex that were projecting more to ventral areas of hippocampus, and in fact, Greg Quirk has recordings and Pat Sharp have recordings that both probably were grid cells that were just really large scale grid cells. And it wasn't until the crucial thing was that Menno Witter said, well, let's look in the region of entorhinal cortex that projects to the dorsal hippocampus, where those, place cells were, the small place field place cells were being shown. And that's what that was what prompted the Mosers to go and record in the dorsal regions of the entorhinal cortex, and part of the reasons that the people weren't recording there was it was right under a big, sinus, filled with blood, and underneath the kind of the ridge of the skull, so it was hard to get at. But once they actually went and looked there, that's when they saw the grid cells that had the smaller, more narrowly spaced firing fields. And it's kind of, it's kind of sad that could have been done. The technology was available to do that 30 years earlier. So people could have discovered grid cells with the technology that was available in the 1970s. They just didn't happen to follow the anatomical connections into the dorsal entorhinal. So I guess it's true that this work is bridging out, but it's kind of surprising how slowly it's happened for bridging out from it.
You
So in
mentioned,
those days, lots of people knew neuroanatomy. Maybe they disregarded it, but they knew it. Now, very few people even know any neuroanatomy. Is that going to slow down our progress?
Yeah, that's a scary thought. I mean, for a while, people were talking about this kind of reduction in number of people working on neuroanatomy. And it is scary because people do need to know the connections. And I would argue against naming areas, things like, fusiform face area or occipital place area, because that's giving this kind of functional naming that's, kind of ignoring the anatomy. And I think people have to stick with the anatomy. Though somebody recently just said that there's been this kind of new, I don't know, rebirth of anatomy, maybe with the sort of the fluorescence tracing and so on. So hopefully it's going to have a resurgence.
You
So,
mentioned,
oh, I'm sorry. Go ahead.
no, you go ahead. I'll come back to it. Go ahead.
So, are you sure? Okay. So, following up a little bit on what Charlie said and the evolution of thought. I mean, this is a very general question, and I want to know your thoughts. Like, we and the models we discussed try to explain grid cells or how boundary vector cells or whatever. But I was thinking more in terms of episodic memory. What you think the function of grid cells and place cells is, because we know the medial temporal lobe is really important to form representations of new contexts, but based on what we were talking today in relation to my own data, we know that when animals become extremely familiar with an environment, and there are lesion studies that prove that, then the medial temporal lobe is not really necessary to navigate contexts in which you have complete familiarity. So what do you think the role of these cells is in terms of encoding episodic memories? I have some ideas, but I want to know your viewpoint because you're an expert on this.
Yeah. I mean, it's important to point that out. Sometimes people forget that, patient HM could navigate familiar environments. He couldn't do unfamiliar, but he could find his way back to where he had lived before he had the surgery. So it really does suggest that the entorhinal cortex and hippocampus are specifically important for forming representations and plotting trajectories in novel environments. And in my book, I laid out how that could happen, how I have a circuit between the grid cells, in this case, grid cells driving place cells, just the way Francesco was modeling it. And then the place cells would be driving speed modulated direction cells. And I was assuming movement direction cells that then could, drive the grid cells in a loop. And you could actually have a full trajectory getting encoded. And the crucial encoding was actually between the place cells and the speed modulated direction cells in that model. But the circuitry there, the grid cells and place cells and speed modulated direction cells in that case are providing a mechanism where synaptic modification can then store these trajectories relatively rapidly. And that's still kind of my framework. And we used it actually in the, Erdem and Hasselmo papers. We actually have the same kind of framework for planning trajectories through the environment. So in the Morris water maze, being able to learn a platform location and then go to any arbitrary location in the space along the perimeter of the tank and be able to plot a trajectory to that location. So to have this extra flexibility of not only remembering trajectories, but plotting new trajectories through space. So that's that's what I've modeled so far, but there's probably additional nuances, particularly with regard to the fact that these systems are malleable based on context, so we know place cells aren't the only thing. There's what we call splitter cells and all sorts of other, changes in the representations that can take place. So the malleability is important beyond what I was able to model in the models in my book.
So essentially, because normally episodic memories happen in contexts that we are extremely familiar with, like in our own house, right? We can make cookies and we have to remember the recipe and how long we cook the cookies or whatever. So you think that the function of these cells is to provide the trajectory and time where these events get encoded, even in familiar contexts.
Yeah, and that's an important question. And I haven't modeled it as much as I should. I tend to focus on completely new circumstances so that you don't have to worry about how the learning is interacting with pre-existing representations.
That's
But
what really troubled me, because I think that most of our episodic memories don't happen in novel. In novel context, you are trying to figure out where you are, but episodic memories happening in scenarios where we are very familiar with the spatial coordinates. It's just that we have to encode all these events that happen in the familiar context. So,
well, I would argue that they're happening in both. I think, the episodic memories that you form in a new city are an important element for probably building up some map over time that, involves consolidation processes. But that is more what I tend to model. And you're right, a lot of episodic memories are, the importance of being able to distinguish between what you said to somebody, like when you come into the office in the morning and you say hi to, a friend of yours, and then when you see them 10 minutes later, not to say hi to them again, or, not to greet them as if you just saw them. Whereas the day before, when you first saw them, you'd have to say hi and then not, so it's really a tough mental task to discriminate between different episodes in a highly familiar environment. In fact, that's what the delayed spatial alternation and delayed non-matched sample tasks, that's partly, I think, why they're so sensitive to hippocampal lesions, is that they're really challenging the animal in a highly familiar environment to remember what it did most recently. So that is a very important question. And maybe, in some ways, well, the ability to set up new contextual representations in the hippocampus is probably a very important characteristic beyond the place cell representation. There's this flexibility, and like we showed with the data, you have these splitter cells kind of popping up, several of them each day, and maybe that's an important part of the process, that you're not locking yourself into a given representation indefinitely, but new cells are coming up and forming new contextual representations every single day so that you can form these new episodic memories.
Well, that's what is suggested by the fear conditioning and reconsolidation theories, right? That each time that you retrieve the information, the context changes, and then you re-encode because time passes by. So essentially, the time variable is not going to be the same, and that changes your perception of the context.
Yeah, and I just want to mention, this is what Mark Howard was really starting with before he got into the modeling of the time cells. He had this temporal context model, which was doing exactly that, where the retrieval of memories was being merged with new memories to form a new context that would evolve over time.
So in listening to you guys talk about that, it almost seems like time is almost it's just a trick, right? Because the time representations that we have here, they're useful in one sense in building space, in building our sense of space, and then later they become useful in building context. So there really isn't a sense of incremental time that allows us to position things. I mean, well, how do we think about that? Because there's many different aspects to time, and all of these things are sort of tools that build something else that's other than a sense of time.
Yeah. So so it's not clear that absolute time is being represented in neural circuits, and that's where the temporal context model was exciting, in the sense that it was representing time by the evolution of the process of context coming in. And this interesting, he developed that model partly to address the fact that if you have a serial position curve in episodic memory tests in humans, that if you give subjects a bunch of words at 2-second intervals, they'll have a certain curve, for 10 words. And if you give, a bunch of 10 words at 10-minute intervals, you get a similar curve of the retrieval of those. And so it seems like the system has this capability of, in a sense, mapping time on multiple different scales with similar characteristics. And maybe the evolution of time, the subjective evolution of time, is dependent on the number of new pieces of information you're getting per unit time, which does, of course, cause all sorts of subjective effects when you're in a new environment versus in a familiar environment, or even going to a new place once versus, you know, going to the same place again, you'll often say, oh, that seemed really different in terms of the time duration that it took. So, and there's other ways of distorting it. So, you know, it's been shown that modulating dopamine cells will change the subjective perception of time for animals. And that's consistent with, you know, some drug effects that have been described in, you know, behavioral tests with, you know, amphetamines, for example. So time is not, you know, it's not clear that there's any code of absolute time.
Now, I would like to raise an issue which I don't think it gets talked enough in our field. And in these studies, usually when we talked about encoding of time, usually, I mean, I divide these studies about roughly in a couple of different paradigms. One is, okay, you have an animal that has a delay, there is a delay epoch in one experiment. And so you start measuring what cells do from the beginning of that delay. And so that's repeatable. And you can make multiple trials and check that the cells are doing the same thing. But then together with this, which I think is a very nice paradigm, it kind of gets conflated with this idea that maybe drift or spontaneous remapping in hippocampus is somehow an encoding of time, which could very well be the case, but it's a very different conceptually, it's very different. And I argue that in terms of classic neurophysiology, you cannot even talk about a neural correlate of time in the latter case, because the idea of having a neural correlate is that you have, you know, a stimulus or a situation, an event, and you measure the cell response, and then you have to repeat that multiple times and show some kind of tuning of the cells or some selectivity. The latter case, you can't repeat time. You know, in the first case, you know, you have a delay, so paradigm, so you can repeat that many times. You know, it's lapse time from a starting point that is experimentally defined. But in the second case, I think it's very vague. So there is so many things that drift in life, you know, in biology, and all of a sudden, of course, they correlate with time. And now, you know, so unless you cannot talk about, I think, a neural correlate of time in that case, and people do, and I think I have a problem with that. But I think the proof is going to be in somehow figuring out a readout mechanism that use that information. Then you're going to have to prove that that system was purposefully encoding time.
Yep.
So that's very important, and I don't think it's discussed enough.
Yeah, no, it's not. I mean, I have to say, you know, really, there's a little bit of a mismatch in the way I present things, in the sense that I start talking about episodic memory and coding of time for episodic memory, which is this long-term representation where no episode is ever happening again. You're just trying to retrieve it later. And so that's almost by definition impossible to study unless you can, you know, exactly measure the millions of neurons involved in reactivating the prior episodic memory. And so, yeah, we're making this kind of oversimplification, looking at tasks where the rat is running for 16 seconds and you see the time cells reading out. But in between, I mean, I'd like to mention, in between, we have found something, which is, it's very hard experimentally to look at a delay that's longer than 16 seconds. But in our data with the imaging, you know, the work that Will Mao did, and actually some of our other imaging data, we at least have the same cells on different days. And so now we can look at not only what happens within a 16-second interval multiple times within a day, but you can look at the sequence of trials, trial 1, trial 2, trial 3, trial 4, trial 5, and see evolution over those. And then the next day, we actually have evidence that there is a correlation structure in which trial 1 is more similar to trial 2 than it is to 3, 4, 5. And so the correlation structure is changing across trials. So at least on a minute time scale, we do now have evidence that there's a similar evolution on each day. It's still not episodic memory because it's not one event on one day. It's how are you forming a temporal structure for, oh, this must be trial 3, you know, whether you're encountering it on day 1 or day 4. But at least it does give evidence for, you know, a longer time scale representation.
Yeah, I agree.
I agree. The time is a hard one for experiments. I think, you know, experimenters should say time may change me, but I can't trace time.
Exactly.
Speaking of time,
Sorry.
do you have anyone else?
Yeah, I don't know how much time we have, but
We have about 10 minutes.
Okay, so I mean, one thing I wanted to point out is, you know, in the beauty of you know, Mike's work. I mean, it's not just Mike, you know, as he mentioned, Neil Burgess and the O'Keefe group, but you know, I think Mike did so much work also in this. Is when I try to summarize it to, you know, people not in the field, is like, I think it's just a way of turning oscillations in time into oscillations in space. I mean, if you really look at the essence of these models that, you know, he talked about, that's what they do, you know, with this trick of velocity modulation and everything. And I think that's, I want to say that's fascinating, because, you know, when we talk about oscillations, you know, they're so omnipresent, ubiquitous in nature, in biology. I think that's even regrettable that it's not very well represented, for example, in undergraduate curricula. I mean, you just get, you have this book, Art Winfrey, The Geometry of Biological Time. I mean, it's just amazing. You know, how you have oscillations everywhere. And in the brain, you know, you might have these oscillations in time that get turned into a representation of space, which is a cognitive function. And so in a way, this idea of time and space, I mean, you know, they're really, it's hard to disentangle them when you, and I think, you know, Mike's work is really, really good at showing that in both experimental and computational work.
Well, thanks. Yeah, I've been excited about the, you know, the use of oscillations and phase coding for years, and I still think it hasn't fully realized its potential. You know, I mean, when theta phase precession was discovered, that, you know, I was talking with Francesco about this earlier. I think that's one of the most important discoveries in our field, because it's such robust evidence and reproducible evidence for, you know, temporal coding, and so robustly, you know, coding the spatial locations. And, you know, you can, in addition, see how that could be expanded to other dimensions. You know, so I had one paper where I said, well, you know, it's not only, it could be used for not only describing the location of the agent itself, but also the agent's perception of the location of other objects in the environment, or even, you know, more abstract concepts like how much has a balloon been inflated, or, you know, how much has the color of a stimulus changed, you know. So there's many dimensions that could be coded by phase coding. And I keep thinking that, you know, if we just look closer and closer at more and more, you know, phases relative to different frequencies, maybe we could find the, you know, code for, you know, much more sophisticated codes for a much more broad variety of stimuli in the environment. So I think there's a lot of potential in that domain.
We have lots of trainees listening, so
Good.
Point taken. Thank you for joining us, Mike Hasselmo. This has been great. Thank you, everyone. This has been Neuroscientists Talk Shop.
Thank you.
Thanks, guys.
Thanks very much. Great
Good.
conversation.
Yeah, this was really good.
That was good.
Who did that line? I was trying, is it Beatles?
No, it's the David Bowie.
Yeah.
Change.
Oh, David Bowie. Yeah, geez, I just could not, I could sing it, but I couldn't come up with the name.
Maybe we'll play in and out with, although that's probably I probably, it's copyrighted. I can't use it.
Sure.
Oh yeah, that's the problem. Have to write your own song.
Thank you.
Thank you
so
again.
much.
And I'll send,
All right.
I'll send you I'll send you a link when we have it up. Sorry to talk over you, Isabel.
No. Thank you, Salma, for organizing it.
Thank you. Bye,
Yep.
guys.
Thanks.
You, Mike. Thanks a lot.
Yep, thanks.
It was super.
Appreciate the invitation. Thank you.
Bye.
Bye.